Hi, I'm Shrek. I'm an associate professor in the Molecular Neurobiology Laboratory at the Salk Institute. And in my laboratory, we use the invertebrate model 𝘰𝘰𝘮𝘦 and the vertebrates zebrafish and mice to study how the brain generates behavior. So today, you're going to hear from three of our fantastic scientists. You're going to hear from Molly Matty, who's a postdoctoral fellow, and research assistants Ahana Chakraborty and Janki Patel. And they're going to tell you about the experiments that they are doing. And once you've seen those sections, you would also have a chance to ask them a couple of questions at the very end. So I think it should be a fun video and a fun interaction with us. At the same time, we look forward to hearing from you.
Hi everyone, my name is Janki Patel and I'm a recent college graduate and now a research assistant in the Chalasani Lab at the Salk. I knew I wanted to study the brain since I was a very young kid. My grandpa would always say, your brain is your strongest weapon, and for some reason that stuck with me all this time. Following my neuroscience ambition, I worked in a neurobiology lab as an undergrad, and that fueled my already bright fire. Today, I'm going to show you guys how I can make cells, which normally sit quietly, respond and glow to ultrasound. I am going to give cells a piece of information in the form of DNA, which will make the cells glow when I turn an ultrasound stimulus on. So let's get started. These are some human embryonic kidney cells, and they live in a tea flask because it's shaped like a tea in the incubator. I'm going to now move the cells from the tea flask onto this small circular glass so I can easily carry it to the microscope for imaging on day 3. I want to put about 300,000 cells on each glass plate, so I'm going to count how many we have in a mill using this little fancy machine and do some quick math, which sometimes can be very difficult. Here we have a 12-hour plate and I've put a glass plate in each of them. After putting 300,000 cells in each plate, I will give them some food, which is this red liquid, and we'll put them back in the incubator. So this is the next day. The cells have gotten to know their environment and are ready to get to work. I'm going to give them all the information they need to make a protein that will get them to glow in response to ultrasound. This information is delivered in the form of DNA. We will give them a day to make the protein, and tomorrow they should be ready to image. I am gently loading the cover slip onto the holder. The transducer is sitting right below this. And because the cells love this red warm media, I'm just going to pour some over them. All I have to do now is hit trigger, and in about 20 seconds we'll see the cells jump a little bit. A better understanding of all this will allow us to develop therapeutic tools for treating neurodegenerative diseases and many other diseases like diabetes and chronic pain that affects a large population of the world. This is very exciting and makes me jump out of bed every single morning. And of course, I absolutely love the people I work with every single day. They're so smart and supportive, and we have so much fun doing cool science every single day. And I'm so grateful for all of that.
Hey guys, my name is Ahana Chakraborty and I'm a research assistant here at the Salk Institute. So as a research assistant, I have a lot of responsibilities, and those include coming up with new ideas for experiments and ideas for projects, as well as assisting others with their current projects that they have ongoing. Part of my job as a research assistant here is to be a member of the Sono team, which comes from the word Sonogenetic. In that project, I'm trying to understand what frequencies of ultrasound certain cells will respond to. In addition to my job as a research assistant, I'm also lucky enough to actually have my own research project, which involves trying to understand the genetic foundation of autism spectrum disorders. So today what I will be showing you in lab is a procedure that we use very frequently for multiple experiments. It's called immunohistochemistry. So that's a very big word, so we can break that down. Immuno comes from the use of antibodies. Histo comes from the actual tissues themselves, so maybe a brain or a different organ. And then chemistry, of course, stems from the word and use of chemicals in these experiments.
So here we have a brain from a mouse that we stimulated with ultrasound. We want to see how the ultrasound may have affected its neuronal proteins. I'm going to freeze the brain now in this little box so that I can cut it into thin slices later. This is our cryostat. Cryo comes from the fact that the inside of this machine is kept at -20 degrees Celsius to keep the tissue intact. I'm going to cut the brain into thin slices of about 20 micrometers. So how does antibody labeling work? When our body fights an infection, it makes antibodies, which recognize foreign particles and mark them for destruction. Let's say we want to label some animal tissue. First, we label the tissue, which is known as the antigen, with something called a primary antibody. Then we tag the primary antibody with something called a secondary antibody. This antibody has a fluorophore attached to the end, which is a fluorescent molecule that emits a certain wavelength of light when stimulated by a different wavelength of light. Now back to lab. Here I am very carefully putting the tissues onto the slides. The slides are ready to cover and seal. Now we're going to turn on the microscope, shine the laser, and ta-da! Now we can see some proteins.
There's so many things that are really exciting about science, so many things to learn and discover. And the more I learn about science, the more I want to go out and do science. The more I do science, I want to learn even more about science. And sometimes you get results that aren't necessarily what you expected. They're quite different from what you expected. And that might feel like a failure, but the good thing about science is that actually nothing is really a failure. You get one result and you take note of it and you add that and you adjust your question. You come up with a better question maybe, or a different question even, and you pursue that next. So there's never a failure, there's always just a motion forward, discovering something new.
Hi, I'm Molly Matty, and I'm a postdoc in the Chalasani Lab here at the Salk Institute. A postdoc is kind of like even more school after a lifetime of school. After my PhD, I knew that I wanted to keep learning and I wanted to keep doing science because my eventual career goal is to work at a college training undergraduates on how to do cool research, how to think through research problems, and teach science classes. One of my favorite parts about my job is probably that I get to mentor undergraduates and just solve problems. I know it seems kind of hokey to like to solve problems, but I just sometimes see a connection somewhere and want to be able to directly interrogate that connection and see if it's real or see what's the cause of that connection, and that's what I get to do in science. So what I'm going to show you today is an experiment that I do in the lab quite often, and it helps me answer the question of what is going on inside an animal as it makes decisions. So let's say that you and your friends in a post-pandemic world are at the mall. You know at the food court there's a taco shop, and you really want that taco. But now let's say that there's a pit of snakes between you and the taco stand. Would you go across that pit of snakes to get your taco? We can ask this question in a much simpler way using a model organism called 𝘰𝘪𝘀𝘀𝑗𝑠 elegans, or C. elegans, which are a 1 millimeter long worm.
So here you can see the experimental setup for the sensory integration assay. We will have assay plates that have a diacetyl odor on them, which is a popcorn smell that worms love. There's also copper sulfate in a line that has been dripped down and dried into the middle of the plate. This is a repellent. The worms hate copper.
So what we can do in this experiment is ask what is normal wild type. How often do they decide to get to that flavor, that compound, that taco that they really like, even when there's something really scary preventing them from getting there.
So I'll place down a population of worms, typically about 100 worms, and then I wait 45 minutes. I then look at the same plate and observe where the worms are. We want to be able to quantify or count or measure these results. To do this, we use a number called the chemotactic index. Chemotaxis is the movement toward an attractive stimulus or away from something repellent. The way we calculate chemotactic index is we count the number of worms on the side with the odor and divide that by the total number of worms that are on that plate.
What we see is usually wild-type animals that have been well-fed and not stressed their entire lives will only cross that barrier to get to that smell that they really like about 20% of the time. Now, if we overly stress the worms by perhaps starving them from food depriving them, then we see that the animals will cross the copper barrier, that toxic repellent, to get to the thing that they want almost 80% of the time. I really like talking about this assay because it lets us realize that we are a combination of not just our genetics and our environment, but also our internal states. So what's going on inside us, biochemically, metabolically, neurologically, what's going on inside. What I hope to do with these worms in this super simplified model system is really understand bigger biological questions like how do we make decisions and what controls choice.
Thank you so much, and I hope you enjoyed our video. Good luck with everything. Bye.